Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
Today we are going to discuss about the direct cell-cell contact. How exactly the cell-to-cell contact happens directly, or if maybe there might be a gap or there might be a junctions or whatever it is from cell to cell, how this particular junction, the signal transaction happens, transformation happens, that we will see. So cell exhibits a special proteins on every cell has got a special proteins on their cell walls that we know. As a biotechnology students, you will be knowing. We will call it a surface antigens, right? So these cell exhibits the special proteins on the cell. Every cell exhibits a special proteins on their cell surface, which is leading to the direct cell-cell contact. Usually these surface proteins or the special proteins, they will be highly specific for cell-to-cell contact. So this type of interaction is highly specific and direct communication between two cells. So however, both soluble signaling and ECM signaling, if you are talking about the soluble signaling and ECM signaling, with respect to the which are different from the special proteins on the surface of cells, so these are non-specific. Okay, so both the soluble signaling and ECM signaling are non-specific, since there is no direct knowledge between the signaling cell and direct cell. But in direct cell-cell contact, what happens is the special proteins will be there from one cell to the another cells. So there will be a proper contact will be there. So there are two forms in the direct cell-cell communications. That the first thing is cell-cell signaling molecules are two different forms. In the first form, what happens? The signal allows mechanical contact between two cells. Okay, so here mechanical contact is there. That means one cell is attached with another cell. That means surface cell or other things like that. The second one is molecules that form junctions. So either first one was a direct mechanical contact. The second one, in between cell A to the cell B, there will be a gap or a junction. They form a junction between the cell through which the molecules can be transported from one cell to the another cell. Okay, understanding? The first form is the direct mechanical contact. The second form is the junction will be there, so that from one A to the B, cell A to the cell B, the molecules will be transferred from whatever the molecules has to transfer from A to B, that will flow through the junctions, okay, from A to B. So that is the second form. So two forms. So there are four different junctions facilitating the direct cell-to-cell interactions in animal epithelial cell tissue. So now this is very simple. Mechanical contact will be there from cell to cell, which are one cell, cell A to cell B. They are coming to contact together. That is done. So whereas in second, for a form where they form a junction between A to B, we have four types. The first one is a tight junction, where this can form an impenetrable barrier from sealing the neighboring cell membrane together. That means there is an impenetrable barrier will be there. Okay, they form impenetrable barrier by sealing. They will seal the neighboring cell. That means the next adjacent to its cell, whatever the cell is there, they will seal that their cell membrane together. So thereby it can prevent the leakage of molecules between the cells. If any molecules are there are transferring from A to B, there should, those molecules will not have any leakage in between these two cells. They are usually forming a barrier, impenetrable barrier, sealing the neighboring cell. That's why it is called as tight junction. But here they are not directly coming to like as a mechanical support or the mechanical contact, that won't be there, but still they are tight junction. That's why they are called it as a tight junctions. So we can prevent any molecule leakage in between these two, cell A to cell B, when they are moving from A to B. So that was about tight junction. The next subtopic is sub junction, just next junction. This thing is, we are having belt desmosomes. In that belt and under desmosomes, we are having belt and spot. So belt desmosomes, as the name suggests, this is called as adherent junctions. The junctions will be there in between the cell A to cell B. So this can join an actin cytoskeleton bundle of one cell to the similar bundle of the adjacent cell. So there will be actin kind of structures will be there. So from one cell that will bind to the same structures of adjacent cell. So this can form a circumferential belt of adhesion. Okay, so that we will see in the diagram. So this is how it looks like, adherent junction. Okay, actin filaments like this from one cell to another cell. That we will see later in the everyone. So next, spot desmosomes, also called it as a spot welds. So here this attaches the tough intermediate filaments in one cell to the another cell. So this is how it looks like. See, a kind of what you say, weld junctions. Okay, here there will be a connection from one cell A to the cell B. So that's why it's called intermediate filaments. So here this can be the intermediate filaments. Here also you can see. Okay, so next we are having the gap junction. So this can form a hollow channel between the adjacent cell, thereby allow the passage of small cytoplasmic molecules across the membrane. This will be the hollow channel like this, for example. So from the starting I will tell you. So for tight junction we can say that example is there. This is the tight junction. Okay, so where the tight junction is this. Okay, from one cell to another there is a compact tight this thing is there, so that there is no leakage from cell A to cell B. Next we are having the next topic was belt desmosome. Belt desmosome, here as the name itself indicates, adherent junctions will be there. So here are the adherent junctions. So from the filaments from cell A to the cell B. And next we are having the spot desmosomes. Okay, small spot desmosomes uses intermediate filaments from A to B. This is how it looks like. These are the filaments of one place and the filaments of another place. Okay, and that's how they welding kind of structure will be there. So that's why they are also called as spot welds. Next we are having gap junctions. So this can form a hollow channel between the adjacent cells, thereby allowing the passage of small cytoplasmic molecules across the membrane. So this is the connection what you see here. So direct connection will be there. So the molecules will be allowed. Okay, that is called as the hollow channel connection. Okay, from adjacent one cell to the adjacent cell. Understanding? So here the tight junction is explained, and here all other cell to cell interactions explained. The desmosomes, the belt desmosomes, the spot desmosomes, and finally the gap junctions. Okay, this is the gap junction. So that was about the 4 different types of the second form, that is where they form a junctions between the cell through which the molecules can be transported from one cell to the another cell. Understanding? So very simple to understand. Next we are having cell junction. Okay, so that particular cell junction, whatever we have the second form uses, that we are discussing separately. Okay, so here molecules, for what you call it as a cell junction, so molecules involved in direct cell contacts are known as cell junction molecules. For example, here actin filaments, okay, and here we are having intermediate filaments, and here connexins. So these all things will act as what? The cell junction molecules. So molecules which are involved in the direct cell contact are known as cell junction molecules. If one cell is directly mechanically in contact with another cell, then that is a different form. And when you are talking about the second form, which is totally pure based on the junctions, that means there is something between the cell A to the cell B. So that between is cell junction. In that between, what are the things are there? What makes them connecting? Okay, so from A to B are called as cell junction. So molecules which are involved in direct cell contact are known as cell junction molecules. Understanding? So this junction may on the order of the around 1.5 nanometer diameter. So this whatever the junction is there, now here this gap junction. So this junction from one cell A to cell B, the diameter, okay, the gap, okay, can be of 1.5 diameter typically, allows the molecules less than 1000 daltons to pass through. That means 1000 molecules can pass through this particular cell, through this junction, which is 1.5 diameter. So assume that the molecules being exchanged. Now with the calculations we will see how exactly the molecules, how many number of the molecules can be transferred, all those things. So assume that the molecules being exchanged, that means whatever the molecules are transferring from the cell A to the cell B. Understanding? Assume that the molecules being exchanged never leave the cells. Okay, what just before the transportation. Okay, never leave the cells, and their size is limited by the diameter of the pore. Okay, now consider the flux through the hole on the flat wall. Okay, there is a flux in the sense what means how speed these molecules are moving. So the maximum flux J is calculated as J is equal to, there is a standard formula for that, 4D divided by pi small d into C1 minus C2. So where D stands for the dimension coefficient, okay, sorry, diffusion coefficient, because the molecules are diffusing from cell A to cell B. So capital D stands for what? Diffusion coefficient, whereas small d stands for diameter of the hole. Okay, so here it is in the denominator. Okay, and C1 is definitely the solid concentration in the signaling cell. That means in our example it is cell A. How signaling concentration, solute concentration in signaling cell. Signaling cell is what in our example from the beginning? Cell A is a signaling cell. Cell B is the receiving cell, right? So C1 is nothing but how much concentration of this particular solute. For example, what is moving is important. Okay, so just before movement from cell A to B, you need to calculate its concentration. That concentration you can call it as a C1. Okay, and then after the transportation, okay, once the receiving cell, the cell B receives this thing, after they pass through the junction and then they receives, that will be C2. That will be the solute concentration in the receiving cell. Got it? So when we substitute the typical numerical values which are commonly in use, that is usually the diameter there we have mentioned is 1.5 meter, that is the starting range, that's as minimum as this thing, and we can keep it as a normal value as 4 nanometer. D is equal to 4 nanometer, and capital D, that is the diffusion coefficient, okay, which is almost a kind of constant, that is 10 to the power minus 5 centimeter square per second, and whereas C1 minus C2, consider it as a 100 micrometer. These are all assumptions, okay, and most widely used values. Okay, so 100 micrometer, micromolar, sorry, concentration. No, so here what happens? The concentration 100 micromolar. So that means in C1, imagine 200, and C2 100. That means only 50% of molecules diffuse from C1 to cell A to cell B. Okay, so now we obtain the flux of around, once you substitute all these values into the above mentioned formula, 4 into 10 to the power minus 5 divided by pi into 4 into 10 to the power minus 9, because it's in nanometer. Nano means 10 to the power minus 9 into C1, that is C1 minus C2 is 100, right? So directly you can call and substitute here as a 100, or 200 minus 100, that will be 100. Okay, so now we obtain the flux of around, so the once you substitute and do the calculation, the value will be 2.4 into 10 to the power 5 molecules per pore per second. That means in between the junction, whatever the molecules are there, that the junction is there, that junction is made up of holes or many pores. Okay, and every pore at every second, how many molecules are passing, that is called as we are having flux. So according to the example or the assumption what we made and what we took, here we can conclude that in every pore, okay, we can from cell A to cell B, 2.4 into 10 to the power 5 molecules can be transported from cell A to cell B within a second, per second. Understanding? That means 2.4 into 10 to the power 5 molecules are transported through the single pore in a single second or per second. So therefore, for 100 pores, we obtain the flux of 2.4 into 10 to the power 7, right? How? Because here we are talking about 100 pores. This is per pore, right? For 100 pore, you have to just multiply with the 100. So when you multiply 100 with 10 to the power 5, 2.4 into 10 to the power 5. This multiplication will simply be the addition of the powers, 10 to the power 5 plus 2, because 2 zeros, just you have to add 2 zeros, 5 plus 2, that is 10 to the power 2.4, just be as it is, and 10 to the power 5 plus 2, because of multiplying factor, that is 100, that is 2 zeros, 2 plus 5 plus 2, 7, 10 to the power 7 molecules per second. So therefore, for 100 pores, we obtain the flux, that 2.4 into 10 to the power 7 molecules per second. That means in 100 pores of a particular junction, from the cell A to cell B, 2.4 into 10 to the power 7 molecules can be transported in 1 second. So that's how we can conclude this particular assumption with the formula. Okay, that is flux formula. So that was about cell junction. So next topic we are having the telomers and self renewal, that we will discuss in the next class.